Hello friends, welcome to our channel. Today we will be discussing about the abnormal neuronal regulation. In the previous video we had discussed about abnormal neuronal regulation, baroreceptor and central nervous system mediated blood pressure regulation. Today in this video we will be discussing about adrenergic receptor and adrenergic hormone mediated neuronal regulation. So in order to understand how adrenergic hormone secretes and it binds to the adrenergic receptor, we need to understand the basic concept of adrenal receptor as well as adrenal hormone synthesis and secretion. So let us understand that from the central nervous system through sympathetic neuronal regulation, there will be a secretion of adrenergic hormones. There are two types of adrenergic hormones. One is epinephrine and second is norepinephrine. Epinephrine is also known as adrenaline and norepinephrine is also known as noradrenaline. Now, from the central nervous system through pre-ganglionic neuron, there will be a secretion of neurotransmitter called acetylcholine. This acetylcholine is common for sympathetic as well as parasympathetic nervous system. It will be secreted from the pre-ganglionic neuron. Let's say for example, this is a ganglia or this is a ganglionic juncture. Those neurons which are located before ganglionic juncture are known as pre-ganglionic neuron, and the neurons which are located after the ganglionic juncture, it is known as post-ganglionic neuron. So from the sympathetic pre-ganglionic neuron, there will be a secretion of acetylcholine, and from the sympathetic post-ganglionic neuron, there will be secretion of norepinephrine in the blood. Secondly, there will be a direct action of this pre-ganglionic neuron secreted acetylcholine on the adrenal medulla. This adrenal medulla will then secrete norepinephrine approximately 20% into the blood and epinephrine approximately 80% to the blood. This is how these neurotransmitters get secreted from the central nervous system to the human blood. Now let us understand in detail about the basic pathological mechanism of secretion of this neurotransmitter into the blood. So I have zoomed out this particular neuron here and I have explained that how there is a secretion of norepinephrine in the blood from the neuron. So we all know that the marker tyrosine is an amino acid. This tyrosine is present in the blood. So this tyrosine is transported into the neuron. This is the neuron. I have zoomed out the neuron over here. So let's say this is a neuron. In this neuron, tyrosine will be transported inside the neuron through some sodium dependent tyrosine transferase channel. So this tyrosine will come inside this neuron. This tyrosine is having a structure which is of a catecholamines. So this is tyrosine will be converted into L-DOPA inside the neuron with the help of enzyme tyrosine hydroxylase. Basically for the structure of catecholamine, there is two hydroxy group needed. But in case of tyrosine, only one hydroxy group is present. So in order to make this structure for the catecholamine, there should be hydroxylation, addition of one hydroxy group over here. That will be done by the tyrosine hydroxylase and that will form L-DOPA inside the neuron. We all know that catecholamine, norepinephrine and epinephrine are catecholamines. So in order to get to the structure of catecholamines, there will be essential process taking place inside the neuron, and that is conversion of tyrosine to L-DOPA with the help of tyrosine hydroxylase. Now this L-DOPA will again convert into dopamine with the help of DOPA decarboxylase, whereas this COOH carboxyl group will be removed. So this DOPA decarboxylase will convert L-DOPA into dopamine. Now the problem over here is this dopamine is having some enzymes inside the neuron which will degrade. So this dopamine will be transferred inside the vesicle which is present in the neuron, will prevent the degradation or metabolism of dopamine, because dopamine is one of the catecholamine like norepinephrine and epinephrine will be destroyed by this enzyme called monoamine oxidase, which is present inside the neurons. So this dopamine then transfer back to the vesicle, which will prevent the degradation of dopamine and helps to convert dopamine into norepinephrine. So this dopamine then converts into the norepinephrine, which is main catecholamine, with the help of enzyme dopamine beta hydroxylase. This dopamine beta hydroxylase will convert dopamine into norepinephrine, and then this norepinephrine will be released when the action potential come. When the action potential come, this dopamines will get released into this synaptic juncture. This is the synaptic juncture. It is called synapse. So this particular neuron will release norepinephrine into the synapse. Those neurons which are present before synapse are called presynaptic neuron, and the receptor present on that neuron is called pre-synaptic receptor, and those neurons which are located after the synapse is called post-synaptic neuron, and the receptor which is present on the post-synaptic neuron are known as post-synaptic receptor. Now when epinephrine is released inside the from the vesicle, when epinephrine released to the synapse, this epinephrine has a several mechanism or action. This norepinephrine which is released from this vesicle will then go and bind to the post-synaptic receptor first. And we all know there are two types of post-synaptic receptor, mainly if we discuss alpha receptor and beta receptor. But in alpha receptor, alpha-2 receptor is also located on the presynaptic juncture. So there are some action of norepinephrine. Once it is released from the vesicle, this norepinephrine will go and bind to the postsynaptic receptors first. That is the first utilization of norepinephrine which is released from the vesicle. Second, this norepinephrine will also bind to the presynaptic receptor such as alpha-2 receptor, which is autoinhibitory receptor, and regulate the secretion of norepinephrine. Third is this norepinephrine will then metabolize by the enzyme COMT. This is catechol-o-methyltransferase. So norepinephrine which is not utilized by the postsynaptic and presynaptic receptor will also be metabolized to the inactive metabolites via COMT, COMT, which stands for catechol-o-methyltransferase. Now the fourth mechanism is this norepinephrine will be coming back to the neuron via sodium chloride norepinephrine transferase mechanism to back to this neuron, and here some of the norepinephrine will be recycled and used and transported back to the vesicle, and some of the norepinephrine will be metabolized by the monoamine oxidase. Please remember, monoamine oxidase is present inside the neuron, whereas COMT, catechol-o-methyltransferase, is present in the synapse. So there will be two way of metabolizing this catecholamine, that is via monoamine oxidase and via COMT, that is catechol-o-methyltransferase. This is how adrenergic hormones will be secreted from the central nervous system to the blood, and from the blood this adrenergic hormone will go and bind to the adrenergic receptor. Now let us understand the adrenergic receptor in detail and how adrenergic receptor works. After secretion of this adrenergic hormone inside the blood, this adrenergic hormone will go and bind to the adrenergic receptor. Now let us understand the adrenergic receptor in detail. This adrenergic receptor are basically divided into two types, alpha receptor and beta receptor. This alpha receptor are further divided into two subtypes, alpha 1 and alpha 2, and beta receptor are further divided into beta 1, beta 2 and beta 3 subtypes. Now first we will understand the alpha receptor, that is alpha 1 and alpha 2 receptor in detail. First thing is how this alpha-adrenergic receptor works. So this alpha-adrenergic receptors are having two subtypes, alpha 1 and alpha 2. Alpha 1 adrenergic receptors are located in post-synaptic nerve endings, and alpha 2 receptors are located in pre-synaptic nerve endings. Let us discuss alpha 1 receptor in detail and how this alpha 1 receptor works. Alpha 1 receptor are known as GQ protein coupled receptor. Basically alpha and beta, both the receptor are G protein coupled receptor, but the mechanism and the way of action of this alpha and beta receptor are different. This alpha 1 receptor is one of the G stimulatory protein receptor, GQ protein coupled receptor, which will activate phospholipase C. Now this activation of phospholipase C will convert phosphatidyl inositol into inositol triphosphate, which is known as IP3, and diacylglycerol. Now the conversion of phosphatidyl inositol to inositol triphosphate, that is IP3, will lead to two changes, that is triggering the release of calcium as well as activation of protein kinase C. Now this activation of protein kinase C will lead to phosphorylation of calcium as well as sodium channel. Now please understand that phosphorylation of any channel will facilitate the entry of cations, and because this protein kinase C is activated and doing phosphorylation of calcium and potassium, calcium and sodium channel, it will facilitate the action potential and it will increase or stimulate the action. So by activation of protein kinase C and triggering calcium will then generate action potential, and it will give either stimulatory or inhibitory action. Because alpha 1 receptor is stimulatory receptor, it leads to stimulatory receptor. So this is how alpha 1 receptor works. Again I am revising. Alpha 1 receptor is activating phospholipase C when stimulated. When alpha 1 receptor binds to the epinephrine and norepinephrine, it will activate phospholipase C, which will convert phosphatidyl inositol to inositol triphosphate and diacylglycerol. Inositol triphosphate converts or stimulate the release of calcium as well as activate protein kinase C. Activation of protein kinase C leads to phosphorylation of sodium as well as calcium channel. It leads to generation of action potential, and there will be action. Now what action alpha 1 receptor has, that now we will understand after understanding the mechanism. So alpha 1 receptor are present in the different places in the body. It is located in the vascular smooth muscle in whole body. So whenever alpha 1 receptor is stimulated, it leads to vasoconstriction, and due to vasoconstriction, it leads to increasing total peripheral resistance and it leads to increase in blood pressure. Second, it is located in the iris. So it leads to, when stimulated, it leads to mydriasis. Mydriasis is nothing but dilation of pupil. Third, it is also located on urinary sphincter. Because alpha 1 receptor is stimulated, it leads to relaxation of the urinary bladder. That is why it leads to urinary retention. And because of this action, alpha 1 blocker are used for the management of benign prostatic hypertrophy, where the urinary retention is a major cause. Fourth, it is also present in the liver, where it promotes glycogenolysis. It promotes the breakdown of glycogen present in the liver. So this is how alpha 1 receptor works, and it has a different physiological action in the human body. The second is alpha 2 receptor, which is the another subtype of alpha receptor. But as I said earlier, alpha 2 receptor is not like alpha 1 receptor. Alpha 1 receptor is located in the post-synaptic nerve ending, whereas alpha 2 receptor is present on the pre-synaptic nerve endings, and alpha 2 receptor is auto-inhibitory receptor. Let us understand why it is auto-inhibitory receptor. First of all, alpha 2 receptor works via G inhibitory protein. It is also worked by G protein coupled receptor, but this is not GQ or G stimulatory protein, it is G inhibitory protein. Because it is having inhibitory action. When alpha-2 receptor is stimulated, it inactivates adenyl cyclase. When it inactivates adenyl cyclase mediated cyclic AMP generation goes down. Actually, ATP will convert into cyclic AMP with the help of adenyl cyclase. But because alpha-2 receptor is inactivating adenyl cyclase, it will prevent conversion of ATP to cyclic AMP. So there will be reduction in cyclic AMP. Because there is a reduction in cyclic AMP mediated protein kinase A activation will not take place. Now this protein kinase A activation is essential for the phosphorylation of again sodium and calcium channel. Because cyclic AMP is decreasing, protein kinase A is not activated, there is no phosphorylation of calcium as well as sodium channel, and because of this reason there is no generation of action potential and there is no stimulatory action. Rather, there is inhibitory action. This is how alpha-2 receptor has an auto-inhibitory action. Now, where alpha-2 receptor are present? As I said, alpha-2 receptor are basically present in the pre-synaptic nerve endings. So, it is doing auto-inhibitory function, as we had discussed in the secretion of norepinephrine, where alpha-2 receptor is located in the pre-synaptic nerve endings. Once norepinephrine bind to alpha-2 receptor prevent further release of norepinephrine from the nerve ending to the synapse. This is how it will inhibit the secretion of norepinephrine via auto-inhibitory mechanism. And alpha-2 receptor is also present in the pancreas. Because it is having inhibitory action in the pancreas, it will decrease insulin secretion. Now let us understand the beta receptor. Let us understand the beta receptor in detail. So this beta receptor are basically divided into three subtypes: beta 1, beta 2 and beta 3. Now let us understand how this beta receptor works. As I said, beta receptor is also G protein coupled receptor, but it is having G stimulatory protein coupled receptor action. It is having stimulatory action compared to the alpha receptor, where alpha 1 is GQ stimulatory protein, whereas alpha 2 is G inhibitory protein, and beta receptor is G stimulatory protein coupled receptor. Now what it will do, when the beta receptors are stimulated, it will stimulate adenyl cyclase. Once the adenyl cyclase is stimulated, it will convert ATP into cyclic AMP and the level of cyclic AMP increases. When the cyclic AMP level increases, it will activate or increase protein kinase A. Activation of this protein kinase A will lead to phosphorylation of sodium and calcium channel. It will facilitate the entry of cation, and there will be starting of depolarization and generation of action potential starts, and it will give some effect. Now what effect this beta receptor gives, we'll be discussing in the physiological action of beta receptors. So let us understand beta 1 receptor first. This beta 1 receptor is mainly located in the heart, where when the beta 1 receptor is stimulated in the heart, it will increase heart rate, it will increase contractility, and it will also increase AV conduction. This AV conduction, contractility and heart rate is increased when there is a stimulation of beta 1 receptor which is present in the heart. It is having positive inotropic, chronotropic, as well as it also increase or facilitate AV node conduction. It is also located in the juxtaglomerulus apparatus. Because it is located in the juxtaglomerulus apparatus, when stimulated, it releases renin. Once the renin is released, it activates renin, angiotensin, aldosterone system, and via RAS mechanism, it increases blood pressure. So this is beta 1 receptor. Let us understand beta 2 receptor. Now this beta 2 receptor are basically present in the bronchial smooth muscle in the lungs, where when the beta 2 receptor is stimulated, it leads to bronchodilation. It is having a different action than beta 1 receptor. Beta 2 receptor is having vasodilatory as well as bronchodilatory action. So in the lungs, when beta 2 receptor is stimulated, it dilates the bronchial smooth muscle and it do bronchodilation. Second, it is present in the smooth muscles of GI tract and uterus. Because it is having relaxing action, in GI tract, it decreases GI motility, and in uterus, it decreases labor. So for labor, there is a contraction required. Because this beta Part2 receptor present in the uterus will not stimulate contraction, it will rather do relaxation. It will not help in labor process. It will delay the labor by relaxing the uterus, and will not the uterus will not able to contract. Third is, it is also located in the arteries of skeletal muscle, so where also it do vasodilation. And lastly, it is present in the pancreas, where it facilitates the insulin secretion. So this is the action of beta 2 receptor. And lastly, beta 3 receptor, which is less explored, but beta 3 receptor is present in the adipose tissue, where when beta 3 receptor is stimulated, it induces lipolysis. It induces the breakdown of lipid in the adipose tissue, and that is why beta 3 receptor is a potential target for anti-obesity drugs. Second is, it is present in the urinary bladder, where beta 3 receptor, when stimulated, it will do urinary retention. It will retain the urine and leads to some abnormalities, or it leads to retaining of urine will also helpful when there is a dehydration like condition. So it is having physiological action where it will relax the urinary bladder, and via relaxing urinary bladder it will do urinary retention. So that is about beta receptor. So in this video we had discussed about adrenergic hormones, how they are synthesized, how they are released, and then how they binds to the adrenergic receptor, and when they bind to the adrenergic receptor, how different adrenergic receptor works, via which mechanism they work, and their physiological action. Now let us coming to the pathophysiology of hypertension, where this abnormal neuronal regulation we are discussing, where this adrenergic receptor when become abnormal, or there is a abnormal secretion of adrenergic hormone, these two condition will disturb the homeostasis of blood pressure maintenance. So this we had discussed in this video. I hope you are liking our video, and if you have any query related to the content we are providing, you can write in the comment box, or you can reach to our social media handles. Thank you for watching. Keep watching, keep learning, keep growing.